Hi, today I'm going to continue the discussion I started the other day of how DNA replicates itself, how RNA is transcribed from DNA, and then just continue with translation, how the messenger RNA um, delivers the message of how to make a particular protein based upon the blueprint of the DNA. So, yesterday I did talk a little bit about the DNA replication, and I talked about the complementary base pairs. And I'm going to review that a little bit. I'm also going to review this whole concept of transcription from DNA to RNA. And then I'm going to talk a little bit more about a few more specifics of how that happens. Okay, so this is the same strand that I was talking about yesterday. It's 9 base pairs long, and just keep in mind that a strand of DNA could be thousands and thousands of base pairs, but I just picked a little tiny bit of a strand of DNA and just randomly made it up. So this DNA is always composed of these 4 base pairs, and we talked already about how they always go together. Like adenine and thymine always go together, cytosine and guanine always go together. So if this was your strand of DNA here, this would be its complementary base pairs, and together they're going to make up the DNA molecule. Now you know that cell division happens, and when cell division happens, each cell is going to need the exact same copy of DNA. So it's able to replicate itself. And I talked a little bit yesterday about how the bonds were quite weak and this strand of DNA could basically unzip itself like a zipper. And then each strand is a template for a new strand, because this T will attract an A, this A will attract another T, and so on. So that's DNA replication. Basically, if I filled that whole thing in, what I'm going to end up with is another strand of DNA, and it's going to be an exact replica of this strand of DNA. And here I just filled it in so with the rungs and the ladder, so you can get the idea. Now we have 2 identical strands of DNA. And that is DNA replication. So good, we've got a blueprint, we've got a plan here, but how do we get that plan to the out to the cell so it knows what to do with that plan? That is called transcription, and that is the job of mRNA. So here I did a little picture of a cell. I didn't draw all the organelles, but I did draw the nucleus. And then I just drew a little representation of the DNA here in the cell. And then this little line here, remember RNA is a single strand, whereas DNA is a double strand. So what you're looking at here is this little strand of mRNA, and you'll remember this M stands for messenger, because that's exactly what it is. It's taking a message from DNA, and it's able to travel through the pores in the nucleus out into the cytoplasm, and then this transcription happens, or I'm sorry, translation actually happens on the ribosomes. So the ribosomes have a little machinery that reads the mRNA and can produce an amino acid from that information that it gets. So this is still the same strand of DNA I'm working with here, except now I am going to transcribe this this side of the ladder into mRNA. So A, remember there's no T, there's no thymine in mRNA, so it becomes U. T, its partner is A, C is still G, C is still G, A is U, T is A, G is C, C is G, and T is A. Okay, so this strand here would be this little strand of mRNA here. Now remember, its DNA is much, much longer than 9 base pairs, but that's all I need to deal with right here. Okay, so we have this mRNA and it's traveling out into the cytoplasm, into the ribosomes. So it's kind of an extra slide I didn't need. Oh, it does say that. The mRNA strand can leave the nucleus and travel to a ribosome in the cytoplasm. And then the next step is translation. So that happens on the ribosome. So all I did here is I had this, these nine bases for my mRNA, and I just wrote them out again here. So it's the same code. You might want to look at an animation video of this, just if you're having trouble visualizing this. It sometimes helps to see this as an animation. There's a number of really good ones on YouTube you can watch. So now I have this code. It's from here, and you see it's the same, UAG, UAG, GUA, GUA, CGA. Remember, I just made this up randomly. You notice that I have separated them into groups of 3. And that's how they're read. They're read in groups of 3. And these little triplets actually have a special name, and they're called codons. So each codon codes for 1 amino acid. And you'll remember that amino acids are the building blocks or the monomers of proteins. Okay, so now we have this UAG. It's coding for some amino acid. I don't know which one yet. How does it know? So I don't have the names of the amino acids up here. I just have whatever, like I just have 2 A's for amino acid, and I'll show you how to figure out what the amino acid is in a minute. So this codon here, UAG, is going to attract this little machinery that's holding a specific amino acid, and it's got what we call an anticodon. It's tRNA, with the T standing for transfer. So it's attracting this because you can see that's its partner, right? AUC. So it attracts that one, and that is holding a particular amino acid. Okay, so then that machinery is going to match them up, and what's going to happen is the ribosome is going to read this amino acid and put it together with the next amino acid, which is going to be GUA and then CGA, and it's going to match them up and march them out in a big long chain of amino acids, which is specifically what a protein is. Okay, so what we're looking at here is a strand of DNA, first of all. And again, this is just a random strand that I made up. It's got A's and T's and C's. It doesn't have any G's, but again, it's random. And here I have transcribed this strand of DNA into mRNA. So the A becomes U, again, the T becomes A, the C doesn't become G, but I guess it pairs up with G. A to U again, C to G, and D to A again. Okay, so this is the codon. This is what we are reading when we're looking for what the amino acid is in the codon. The anticodon, just to go back one here, this is what is attracting the mRNA to the anticodon here with the amino acid that is associated with it attached. Okay, so that's tRNA, and the anticodon. Oops, we're good. Okay, so U to A, U to A to U, G to C, U to A, U to A, G to C, A to U, and A to U. Okay, so if the question was then asking you for the amino acids that this would be translating into, what they're asking you for is the codon, the mRNA. So you'd have to find out what the amino acid associated with UUA for this first amino acid, GUU for the second amino acid, and GAA for the third amino acid. So remember, groups or chains of amino acids are what makes up proteins. So now we need to go to the codon wheel. So our first one was UUA. So we would look in the middle here where it says first base, and that's U. Our second base is U, and I've highlighted it here. And our third base is A. Okay, so it's going to be, can barely read it there, but it is LEU, and that's going to be leucine. So our first amino acid is leucine. So again, you just find your first base, so you know it's in this quadrant here. And then your second base, so you know it's in this little slice of the pie, the next one, and then you just follow it through. Let's try the next one. So the next one was GUU. So same thing, because it's G, you know it's in this quadrant. And then the next one is U, so you know it's in this piece of the pie here for U. And then it was U again. And again, that's hard to read, but that says VAL, valine. And our third one was GAA. So we've got G, and so you know again, it's in this quadrant, and it's the A in that quadrant, and then it's going to be the A in this little piece of the pie here. So A, follow that through, and that says GLU for glutamate, so you know that is your amino acid. You can basically do that for any codon that you're presented with. You just go first base, find your second base, and then find your third base, and then just follow it through to get the abbreviation, because they don't have room to write the whole name in here. There's a little table here too, and you could, I guess, just look for it in a table like this, like say it was AAU, you could find that would be asparagine, or GCC, alanine, CCC, that would be proline. You notice that there is a lot of overlap too, because you have these four bases, and they're going to go together in many ways, way more than how many amino acids there are. So you've got quite a bit of redundancy here. Like quite a number of different codons will code for the same amino acid in a lot of cases. You're going to have much more than one. So again, just to summarize that, if we wanted to see what the amino acid, if we were asked in a question what the amino acids were, now we can answer that. Just go back to the codon wheel and follow it through, and you can easily figure it out. And that's it for today.